Welcome to the Deep Dive. Today we're tackling a really fundamental group of biological molecules, lipids. Often misunderstood, I think. People just think fat.
That's right. And there's so much more than just, passive storage.
Absolutely. We're talking about the body's main concentrated fuel source, yes, but also, crucially, the building blocks for every single cell boundary. They're involved in, well, almost everything it feels like sometimes.
Energy, structure, signaling. It's a really diverse group. They're quite heterogeneous, actually, which makes defining them clearly pretty important.
Okay.
So our goal today, our mission, if you like, is to really nail down that definition, walk through the standard four-part classification, and then highlight their essential functions. 5 key roles.
Great. So let's start right at the beginning, the basic definition. What makes a lipid a lipid? It's mostly about water, isn't it?
It really is. Fundamentally, lipids are organic substances that are, relatively speaking, insoluble in water. They avoid it, but put them in an organic solvent, like ether or alcohol. They dissolve quite readily.
Water hating, basically.
Pretty much. And chemically, to qualify as a lipid, a compound needs to be either actually or potentially related to fatty acids. Fatty acids are kind of the core building units.
Right. And this is where they really differ from the other big biological molecules. We hear about proteins, carbohydrates, nucleic acids. Those are usually huge. Polymers.
Exactly. That's a key distinction. Proteins, starches, DNA, they're polymers. Long chains of repeating units. Lipids. They're not polymers. Mostly small molecules, actually.
Which gives them a certain flexibility.
Absolutely. They don't have to form these massive, rigid chains. They can act individually or, self-assemble into structures like membranes very dynamically. It's a completely different structural approach.
Okay, interesting. Not polymers, water insoluble. Got it. So with those ground rules, let's get into the classification. You mentioned 4 categories.
That's right. Based on a system modified from Bloor. 4 main types: simple, complex, derived, and miscellaneous. We can start with simple lipids.
Simple sounds straightforward.
And they are, relatively speaking. Chemically, they're defined as esters of fatty acids with various alcohols. An ester is just that chemical bond formed when an alcohol links up with a fatty acid.
Okay. And within this simple category,
2 main kinds. First, the ones everyone knows, fats and oils. Technically, they're called triacylglycerols.
triacylglycerols.
Right. These are specifically esters of fatty acids with glycerol as the alcohol. And the difference between a fat and an oil, it's purely physical, nothing more.
Ah, okay. So like butter versus olive oil.
Exactly. If the triacylglycerol is solid at room temperature, we call it a fat. If it's liquid, it's an oil. Same basic chemical structure, just a different physical state, usually because of the type of fatty acids involved.
Gotcha. Solid, fat, liquid oil, same family. What's the second type of simple lipid?
Waxes. Still esters of fatty acids, but the alcohol component is something other than glycerol. Usually it's a long-chain alcohol, like cetyl alcohol, for instance.
And what are waxes used for? I mean, beyond candles.
Well, naturally, they form protective coatings. Think the shine on an apple or a leaf. Industrially, yeah, candles, but also lubricants, cosmetics, polishes. They're great for creating water-resistant barriers.
Makes sense.
Yeah.
Okay, that covers simple lipids. What about category 2? Complex lipids.
Right, complex, or sometimes called compound lipids. This is where things get, more complex. They still have the fatty acid and alcohol core, but they contain additional groups.
Like what kind of groups?
Could be phosphate, maybe a carbohydrate, sometimes a nitrogenous base. Even proteins can get involved.
Some more functional groups attached.
Precisely. And probably the most important subgroup here structurally are the phospholipids.
Okay, phospholipids. I know those are important for cell membranes.
Absolutely critical. They must contain phosphoric acid, and often they also have a nitrogenous base attached. It's that phosphate group that's key.
Why is the phosphate so important?
Because the phosphate part is charged, it's hydrophilic, it loves water, but the fatty acid tails, they're still hydrophobic, they hate water.
Oh, so they have like a split personality, part water-loving, part water-hating.
Exactly. That property is called being amphipathic. And it's the secret to life, really. Because of that duality, when you put them in water, they automatically arrange themselves into a bilayer, heads facing the water, tails hiding inside.
The cell membrane structure.
The very same. Without phospholipids, no cells, period.
Wow. Okay. And are there different kinds of phospholipids?
Yep. 2 main types based on the alcohol. Glycerophospholipids use glycerol as the alcohol lecithin, is a common example. And sphingospholipids use a different alcohol called sphingomyelin, found in nerve sheaths, is the classic example there.
Okay, glycerol or sphingosine backbone.
Got it. What else falls under complex lipids?
Next up are glycolipids. Glyco meaning sugar or carbohydrate.
So lipids with sugars attached.
Essentially, yes.
Hmm.
They contain a fatty acid, a carbohydrate, and a nitrogenous base. And like sphingophospholipid, they typically use sphingosine as the alcohol. Sometimes they're called glycosphingolipids for that reason.
And how are they different from phospholipids structurally? The key difference is what's missing. Glycolipids lack both glycerol and phosphate. That makes them structurally distinct. You find them a lot on outer cell surfaces, involved in cell recognition, things like cerebrosides and gangliosides in nerve tissue.
So phosphate means phospholipid, carbohydrate means glycolipid, roughly speaking.
That's a good way to think about it. And just briefly, there are other complex lipids too, things like lipoproteins, which are big complexes of lipids wrapped with proteins for transport in the blood, plus sulfolipids, aminolipids. It gets quite detailed.
Right, a very diverse category. Okay, that's simple and complex. What's category 3?
Category 3 is derived lipids, and this is where it gets really interesting in terms of function, I think.
Derived, meaning they come from the first two groups.
Exactly. They aren't built from scratch in the same way. They are the products you get from hydrolysis, basically, breaking down the simple and complex lipids we just talked about, often using water in the process.
So breakdown products, what kind of things are included here?
It's a really important group. It includes the basic components like glycerol and free fatty acids, which cells can reuse immediately. But critically, it also includes things like the fat-soluble vitamins A, D, E, and K.
Vitamins are lipids, or derived lipids.
Derived lipids, technically. They have lipid characteristics insoluble in water and often originate from lipid precursors. Also in this group are steroid hormones like testosterone and estrogen, various hydrocarbons, and even ketone bodies, which are an alternative fuel source.
Wow, okay. So the breakdown products of, say, storage fats are actually these incredibly active signaling molecules in vitamins.
Precisely. They still have that core lipid property of water insolubility, but they're often highly active biologically, not just structural or storage components.
Fascinating. Okay, that leaves category 4. Miscellaneous. Sounds like a catch-all.
And it pretty much is miscellaneous lipids. It's for a large number of compounds that have lipid-like characteristics, mainly being hydrophobic, but don't quite fit the structural rules of the first 3 categories.
Like what sort of things?
Think of carotenoids, the pigments that make carrots orange, squalene, which is a precursor to cholesterol, various terpenes, even specific long-chain hydrocarbons like pentacosane, which you find in beeswax along with the wax esters. They're lipids, but structurally diverse.
Okay, so a necessary category for things that don't fit neatly elsewhere. Now, before we jump into functions, you mentioned one other term, neutral lipids.
Ah, yes, neutral lipids. This isn't really a structural category like the others. It's based purely on electrical charge. These are lipids that carry no net charge.
So, electrically neutral.
Correct. This includes the mono, dis, and triglycerides are fats and oils, plus cholesterol itself and cholesterol esters. It's just a way of grouping based on charge.
Got it. Okay, so we have the definition, the 4 main classes, simple, complex, derived, miscellaneous, and the idea of neutral lipids. Now, the big question, what do they do? Why are they so vital? You mentioned 5 key functions.
Right. Let's run through them. Function number 1 is the one we started with, the most obvious, concentrated fuel reserve.
Energy storage.
Exactly. Triacylglycerols are the body's primary way of storing energy long-term. And because lipids are hydrophobic, they don't need to be stored with water, unlike carbohydrates. This makes them incredibly dense, more than twice the energy per gram compared to carbs or proteins. Super efficient fuel.
Okay, fuel reserve, function 2.
Structure and permeability. We touched on this with phospholipids. Lipids, specifically phospholipids and cholesterol, are the absolute core components of all biological membranes. They don't just form the boundary, they actively regulate what gets in and out. Membrane permeability is crucial.
Right, the gatekeepers of the cell. Makes sense. Function 3.
Vitamin supply. Lipids act as the carriers or solvents for the essential fat-soluble vitamins, A, D, E, and K. You need dietary fat to even absorb these vitamins properly. Without lipids, they're basically useless to us.
So you need fat to use those vitamins. Okay. Function 4 sounds important. Metabolic regulators.
Hugely important. This links back to that derived lipids category. Lipids act as powerful cellular metabolic regulators. Think steroid hormones derived from cholesterol.
Right. They control so many processes.
They do. And because they're lipids, they can often slip right through cell membranes and interact with internal receptors, sometimes directly affecting which genes get turned on or off. That's incredibly direct control. Prostaglandins are another example, lipid derivatives regulating inflammation, blood clotting, pain. critical signals.
Powerful stuff. Okay, and the last one, function 5.
Physical protection and insulation. Lipids provide cushioning around vital organs, protecting them from physical shock or injury. Think of the fat pads around kidneys. They also serve as an insulating layer beneath the skin, helping to maintain body temperature, and they contribute to the body's overall shape and contour.
Padding, insulation, and shape. Got it. So
Yeah.
fuel, structure, vitamin carriers, regulators, and protection. That's quite a portfolio.
It really is.
It's amazing, really. We started with this simple idea, doesn't dissolve in water, and ended up with molecules that are energy banks, cellular architects, vital nutrient carriers, powerful messengers, and protective padding all rolled into one.
It definitely highlights a kind of duality, doesn't it? Especially when you think about derived lipids, we define them as breakdown products, hydrolysis products.
Yeah, things left over after breaking down fats.
But those leftovers include steroid hormones, vitamins, incredibly active molecules. They aren't just passive byproducts. Their creation is the activation step in many cases.
That's a really interesting point. The breakdown is the function sometimes.
Exactly. So maybe a final thought for you, the listener, to consider is this. We often focus on lipids like triacylglycerols as just storage, the fuel reserve, and cholesterol as just a structural part of membranes or something bad for arteries.
Yeah.
But given that the derived forms, the breakdown or modified forms like steroid hormones, are such potent immediate regulators, how should that change how we think about the balance? The balance between lipids as passive storage versus their role as precursors for immediate, powerful action in the body.
That's a great question. Balancing the image of the stored fuel versus the active messenger derived from it, definitely something to chew on. So much complexity hidden in these relatively small molecules.
Indeed.
Thank you for walking us through that. A fantastic deep dive today.